acids are the single building blocks, the monomers, or the Lego pieces, that when you put together, you get proteins. This is an example of an amino acid, their shape. The R is for the different functional groups that they have. And this is the carbon chain. As we covered last week with functional groups, a different functional group is going to allow it to have a different function because it is a different structure. So for example, here we have different amino acids and their R groups or their functional groups will tell you a different function they might have. So these two, they're hydrophilic, they like water, and this one's hydrophobic, it doesn't like water. When we look at proteins, those are the polymers. They're also known as polypeptide chains. Poly meaning many. So here is an example of a polypeptide chain. Each shape would be a different amino acid. Now, another question about a peptide chain. That would just be a very short polymer. So it might be just one or two amino acids strung together. And what the length of them, well, remember that these are instructions to build proteins. No, sorry. The length of them denotes what kind of protein they're going to be, what their function is. So if they're really short, it might just because they're a small protein, they only do maybe they're just a little membrane channel, whereas some proteins are much longer and fold in to a bigger shape. They have different functions. So their lengths are just dependent on what protein they are and what function they play. So here we have their primary structure. This is just amino acid, amino acid, amino acid, amino acid, just a chain, a polypeptide chain of amino acids. See how they're all different. So it's not just all alanines or all glycines, they're all different, which means they all have a little bit of a different structure. So that's your primary structure, is just the order of which the amino acids are. The secondary structure is when this chain begins to kind of interact with itself. So some amino acids might create hydrogen bonds with another amino acid in the chain. Just like what we've seen with DNA and the double helix, they're made up of hydrogen bonds is what helps twist it. It's the same thing that's happening in this secondary structure. Hydrogen bonds are made between different amino acids, kind of bending or spiraling the chain.
So why do these bonds get made? Like, is it just like a side effect, or?
It's just, so a hydrogen bond is just an attraction that happens between hydrogen and usually oxygen. It's just a very weak bond that happens between the two because of the electron wanting to be shared.
Okay.
After the secondary structure, we have the tertiary structure. And with this is when we start to see some of those functional groups, such as being polar or non-polar, or hydrophobic and hydrophilic, come into play. So this is when it starts to twist into a specific shape based on the different functional groups. And you see this little spiral within it? That's this. So that little spiral that you see, that's the secondary structure. So there's hydrogen bonds making that little spiral or helix, and then different interactions between the other amino acids are causing it to fold a certain way. The quaternary structure is when you have subunits or more than one polypeptide chain come together and make a unit. So this is an example, hemoglobin. They have 4 different polypeptide subunits that come together, and then it's responsible for carrying or transporting oxygen.
So does each protein structure, like, is the first structure within the second structure, and the second structure is within the third, and the third within the fourth?
Yeah, so it's kind of a gradual thing. So the primary structure is all just about the order of amino acids.
Yeah.
So this is actually, you can fill this in. What allows for different structures? Primary structure is the order of amino acids. The secondary structure is some hydrogen bonding happening. So let's say, again, I don't know which ones would do it, but let's say these two, there's a little bit of an attraction going on. So it kind of starts to bend that polypeptide chain, twisting it into this shape. And the tertiary structure, this shape that you see, is right here. But you see other connections happening across the chain. So maybe this one is connecting to this one. So it's starting to fold it into more of like a 3D structure.
Yeah.
And then you get your quaternary, which is your more than one peptide chain. So it's continuous of each other.
And so the fourth one is just like a bunch of them together?
Yes, a bunch of them working together. So sequence of amino acids, that's your primary structure. Bonds between the parts of amino acids, such as hydrogen bonds, is the secondary. Tertiary more focuses on those functional groups. So if they're hydrophobic, hydrophilic, polar, nonpolar, it's all going to change that 3D shape. How is protein diversity related to evolution? So why does this shape matter?
I think the shape has an effect on the function. So if we have changes over time in the genes that are affecting the creation of the proteins, then they're going to have different functions. So there's going to be different like things happening in the bodies of the creatures or the animals and stuff like that.
Yeah, exactly. That big theme of structure, function. So proteins are so different, and even a small change in a protein can cause a different function. I think last year you looked at sickle cell anemia, if I'm not wrong. So in that one, we're looking at hemoglobin. This is a normal functioning hemoglobin. One little change in the amino acid sequence can make this hemoglobin not look like this. Just even that little bit of a change of structure can change its function. Up here, protein structure and function. It's a very reoccurring theme. Here are a few examples of proteins and their functions. Proteins they can do a lot of things in our body. They can be for more structural purposes, like forming the cytoskeleton, transporting materials across the cytoskeleton, cyto meaning cell. They can be enzymes. These help facilitate chemical reactions. So those dehydration and hydrolysis reactions usually need an enzyme to even happen. Channels that regulate the movement across the cell membrane. Receptors on the cell, used sometimes for cell signaling or detecting the environment on the outside of the cell compared to the inside. Also functions such as carrying molecules in the body or storing certain molecules in the body. Hormones are also an example of a protein, some hormones. And a hormone such as oxytocin. That reminds me, my sister is actually being induced right now, so she's pregnant and being induced because she's past her due date, and this is what they would induce her with, is some oxytocin. Also, insulin is another example of a protein. They act as molecular messengers. The last two, antibodies, they protect us against pathogens, and keratin provides a hard waterproof coating in feathers, scales, and skin.
I have a question about enzymes.
Yes.
So it says that the proteins, like enzymes are proteins, right?
Yes. Yep.
Okay. So example, like someone who doesn't eat meat, after a certain amount of time without eating it, they stop producing the enzyme. Is that because the proteins necessary to make the, I know that like the body like stops producing it because it's like, oh, there's no use for it. But could that be because like they're not getting the protein from like a meat, and meaning they can't produce the enzyme to digest the meat?
I don't know for digesting meat, but let's say gluten intolerance.
Yeah.
There is an enzyme that helps break down gluten. And
Yeah.
sometimes that enzyme, it just might have some kind of malfunction where the polypeptide chain is not correct or the folding isn't correct, and that deems the enzyme inactive. It won't work properly. So it might be, I'd have to look up for the meat one, but it could be something similar. Or maybe you don't have enough of the amino acids, so it's not being built correctly.
Yeah, that was more my question. If like the proteins or the amino acid that you get from the food is used to produce the enzyme for that food, but that it's kind of, I don't know.
Yeah, I'll try to look up an example of that, but I know what you're talking about. Like if you're low on a certain amino acid, there you might have side effects with some specific enzymes that might have that amino acid.
Yeah, okay.
Okay. So a little note to do with proteins is that they can be also, so we've seen that they can be changed, their structure can be changed, which would change their function, but also some environmental changes can alter the protein structure. And when we say alter, that could be cutting some of those hydrogen bonds that we've seen in that secondary structure. So if those bonds are broke from heat or really low pH, then we don't have our normal structure, right? It's all disassembled and just like one polypeptide string. It doesn't have its shape anymore. So this denaturing loses or loss of its proper structure means that it also loses its function. So this question at the bottom is asking, how is protein denaturization related to homeostasis? So that, remember, we want to keep the body at a certain level. How would that be related?
Like adapting to changes? to keep the body at level?
Yeah. So we want to adapt to keep the body at homeostasis. Let's say we're talking about temperature. Why do you think we want to keep the body at a certain level?
Because if it gets too hot and our proteins start losing their functions, we'll stop working.
Yeah, definitely. So too hot, and we are going to get our enzymes broken down like this. So that's why we want homeostasis to kick in. We want to keep at a certain level, because once we've reached too high of a heat, then our proteins are denatured, or too low of a pH as well. I've seen some hands raised. Can't see who they are. Was there a question?
Yes, hello.
Hi.
Is a couple degrees like really enough to like actually denature proteins? I feel like that seems kind of like a extreme. I don't know.
Depends on the protein and how like durable it is, but some proteins, they can be denatured quite quickly, even a few degrees.
Okay.
Good question.
Is it possible that proteins don't act unless, like, say in homeostasis, certain proteins don't function or do anything, but only when your body temperature goes up, then they start to work? Or like when the pH goes up or something?
Yeah, sometimes they require certain environmental cues in order to start working. Is that what you mean?
Yeah.
Only at a certain level will it's more so, especially for enzymes, the protein enzymes. Some of them have like a very key, can't think of the word, but like a perfect level where they work best. They're most active at that certain level. Anything lower or higher, they're not going to function at their best.
Okay.
You mean optimum?
Yes, thank you. Where's that word?
Kind
Yeah.
of like if you have I don't know, like pepsin or something in your stomach and it's digesting. Depends on the pH or something like that.
Yeah, definitely.
Okay.
So it has that optimal pH that will work best at breaking down whatever molecule. Okay, on to lipids, our last macromolecule. Their defining characteristic is that they're nonpolar. They have many different structures. And following the theme, many different structures means many different functions. Some of these functions might be keeping the body warm, as you see with this little chubby seal. They are, so insulation, they store energy. They can also be used for cell membranes of organelles. Some are steroids or hormones. So with lipids, they're a little different because they are, so polymer is repeating chains, repeating units, right? Of similar or the same unit. Well, they're a little different because they're not repeating of the same thing. So here we have a lipid. They're attached to a glycerol, and glucose. Is it glucose? So they're two different molecules. Oh, glycerol and fatty acids. Sorry, fatty acids and glycerol. So different units. So really technically maybe not a polymer by definition. So your fats or triglycerides are these. These are what store energy or cause insulation. Phospholipids are these, which means they have kind of a hydrophobic and a hydrophilic layer part to them. So they create these layers. Cholesterol is an example of a lipid. They stabilize the cell membrane. So here's the cell membrane, and there's some little cholesterols in there. They can be steroids, so estrogen versus testosterone, which are molecular messengers. You can also find them as waxes. That is a nasty picture, some earwax. Some waxes for plants to help keep in water. For bees, they use them to store nutrients and protect their larvae. So food for thought, we can take this up on Friday, and I kind of answered it, but do lipids form polymers? And are lipids considered macromolecules? So think about that for Friday, and we'll take that up. So those macromolecules, they're all examples of organic because they had the carbon backbone, carbon, hydrogen. We are now going to go into a few important inorganic molecules, one being water. All life is dependent on it. And there's a few important properties that water have. One is cohesion or surface tension. Example of when we can see water traveling up the tree. So how water interacts with itself. And adhesion is how water interacts with other things, kind of becomes sticky. Also why you can see this bug skim across water. Thermal inertia. So this is the fact that water takes a long time to heat up, but it also can keep its heat very constant. It takes a long time to heat up, but also a long time to lose its heat. This is very important when we think about the human body and how we're made up of water. Keeps us warm. Lubrication and protection.
Wait, what does it lubricate? I don't.
So have you ever tried to swallow a bunch of crackers and it gets stuck in your throat? So your saliva, you need water to help, lubricate down the gullet into your stomach.
I was thinking about like in a machine, like joints have lubrication. Like we don't have that, right? I don't know.
In our joints, we do have some lubrication, which it's not just water in there, but water is needed to make that. But it's not like you were thinking in that way. So more so for making the substances that help lubricate, especially in our digestive system. It's a substrate for chemical reactions. I'm not going to go too much into detail for this because we'll cover it in the next week. And it also acts as a solvent. So if you think about it, a lot of things are able to dissolve into water. And that's a good thing because by doing that, it really helps our body out. If you think about blood, the plasma in blood is used to transport nutrients. And a big part of that plasma is that it's made with water. So it's able to dissolve that nutrients and let it travel, nutrients and ions. Two other important compounds are oxygen and carbon dioxide. Oxygen is the gas that we breathe, so very important. It's also needed for cellular respiration. And carbon dioxide needed for photosynthesis. Anyone know what these two pictures are down here?
Left one I remember from last time. We did like, what's this picture? That's mitochondria.
Yeah, this one is mitochondria. Anyone know this one?
Is that a chloroplast?
Yeah. I know it doesn't have that telltale sign of it being green, but when you watch the capsule, you'll figure out what these little stacks are inside of it. Okay, so for the last activity, hopefully on your sheets you have this. I'm going to break you into groups and get you to think about what you've learned and try to fill it in. And I'll post the answers after class. But I'll break you into groups and try to talk about the functions of each macromolecule and a few examples.